First of all, I would like to thank Professor Roskam for the kind invitation. It is my pleasure to be here with you today. And actually, I apologize for the last minute change in the topic. But after I saw the final agenda, I thought that actually would be more appropriate to provide you with an overview of what we have learned from molecular profiling in terms of mixed tumors and some specific subtypes of HCC on iCCA with stem cell futures. Therefore, I apologize, but I think it fits better with the overall agenda, and hopefully you will think so too. So we have heard already, actually, the great introduction from Professor Roskam, who has introduced the large and complex spectrum of liver tumors. And for this talk, in the next 20 minutes, we will focus only on those arising within the liver, so please disregard extrahepatic or gallbladder cancer. In terms, in particular, of liver tumors, we know that the most common are hepatocellular carcinoma and intrahepatic cholangiocarcinoma, but as we mentioned, there is quite a complex spectrum that is like, it's not always so easy to discern one from the other. So over the years, there has been a large, a growing interest in actually performing molecular profiling of different liver tumors, particularly HCC and intrahepatic cholangiocarcinoma. But more recently, we have seen also novel studies whereby applying whole genome sequencing, whole exome sequencing, and RNA sequencing profile, we have learned more about this unique subtype of mixed hepatocellular cholangiocarcinoma and also cholangiocarcinoma, which is here intentionally separate from the other mixed tumors. We have heard again from Professor Roskam that right now there is general consensus that multiple cells within the liver will actually be considered the cell of origin of these liver tumors. This is still highly debatable. It's not my intention to investigate a debate about this. I definitely have less experience than the other speakers, but just here I wanted to simply represent what we have known till date and what we have learned in the past few years from molecular profiling. Traditionally, we thought, and I think it's well accepted, that intrahepatic cholangiocarcinoma mostly derived from the differentiation of the neoplastic transformation of cholangiocyte, whereas classical or pure HCC derived from hepatocytes. And please, again, I put a note here, this is just a simple representation of these tumors and definitely has no intention, should not be taken as proof of origin. This is just based on molecular evidence of what we have learned to date. So we have pure intrahepatic cholangiocarcinoma, pure HCC, which seems simple, but as we know that it's not so simple. Indeed, we have subtypes of HCC with stem cell future, and also we have seen cholangiocarcinoma with stem cell future. But then where do these fit in this scheme? And what about mixed hepatocellular cholangiocarcinoma? What about cholangiocarcinoma? Definitely, hepatocellular carcinoma has been well studied over the years, perhaps the most well studied of the liver tumors. We have seen a large-scale genomic profiling in the Asia Pacific countries, but also in Western countries. And now there is general consensus that we have two main classes of hepatocellular carcinoma. This is based on molecular profiling, particularly transcriptomic analysis, but also integration with mutations and chromosomal aberrations. And here we know that we have a proliferation class, which is the aggressive HCC. We have different subtypes, but they're all characterized by enrichment of several oncogenic pathways. Some are targetable, and they have a unique methylation profile. More importantly, they're more aggressive tumors. They have high frequency of vascular invasion, high levels of alpha-fetoprotein. On the other hand, we have this non-proliferation class, which is mostly enriched in HCC with mutations in CTNB1 pathway, which have been associated with an immune-excluded phenotype. But what is most important is that within the proliferation class, there is a unique subtype of HCC, which has been defined with stem cell futures. And here, these are positive for CK19, for Icahn. They have a unique methylation profile and so on. But how does this subtype relate to the other? The mixed tumors, the cholangiocarcinoma with stem cell futures. Moving to cholangiocarcinoma, this has been less studied at hepatocellular carcinoma, but similarly, there is general consensus that on the molecular profile of the tumor, we can identify two different classes. This is very similar to the HCC, right? We have a proliferation class, we have an inflammation class. And as the same name suggests, the proliferation class is the tumor that are aggressive, poor survival, several oncogenic pathways, a lot of mutation, and more chromosomal instability. The inflammation class, on the other hand, are well differentiated iCCA with good prognosis. But what I want to draw your attention is that, again, within the proliferation class, there is, at least at the molecular level, there is a group of cholangiocarcinoma with stem cell futures. which is quite unique, in my opinion. I was actually very skeptical. In the past few years, my group has tried to update this classification, actually improve upon this classification. And because over the years, we thought that what we kind of like disregarded was the mu microenvironment. Therefore, we have published, hopefully published, is currently under review. We have proposed a novel classification of intrahepatic cholangiocarcinoma. This is mostly based on immune and stromal features, but interesting. And what was really appealing to me is that once again, although we focus on other features, particularly stroma and immune, we still identified a class, which I will describe in a second, that we call the patistem-like. These are all pure intrahepatic cholangiocarcinoma, but at the molecular profile, we confirmed that these exist. This is a stem cell-like ICCA, again, just molecular profile, but we were able to identify it again. Going into the details of this classification, we performed an alternative classification. We were not interested in the tumor cells. We have performed several molecular profiles in the past of the tumor cells. What we wanted to highlight was the immune and stroma composition of cholangiocarcinoma. Therefore, we did a virtual deconvolution where we isolate the signal coming from the immune and stromal cells, and we did this in 900 samples. And what we identified was a novel classification. Forget about the previous two, which were kind of heterogeneous and were not really providing insights into the details of each subtype. Now we have identified 5 classes. You can see here at the top of this heat map, these are the 5 boxes. Each one was assigned to a specific name based on their molecular features. And here you can see, for those who are not familiar with heat map, just look at red color means high. In terms, these are either gene or signature, so it means that red is high and blue is low. And as you can see, the main determinant of this classification was the immune composition of these tumors. Indeed, we have here 2 classes with really high immune infiltration. And then we go from the left to the right, there is a progressive decrease. And we have this class here in blue, which is the one with the lowest. The one that I mentioned before, the patistem-like, the way we call it, was kind of an intermediate level of immune infiltration. In terms of the 2 inflamed, as you can see, these are really high inflamed. We have a lot of immune infiltration. We've also confirmed at least a pathological level, but they were different in terms of stroma composition. And in particular, this class here that we called inflammatory stroma had a lot of myofibroblastic, high stiffness extracellular matrix, and activated stellate cells. This class that we renamed the patistem-like was the largest, was quite surprising. This is a 32, 35% in the different cohorts. And we observed that clearly overlapped with a previous class of cholangiocarcinoma with stem cell feature. The same that I was skeptical about, because it's like, these are pure cholangiocarcinoma. We did exclude all mixed cases. These are pure cholangiocarcinomas also reviewed by Dr. Swan-Tang, who is the pathologist in all in our studies. And here you can appreciate how they look. This is the patistem-like inflammatory stroma. There was a significant abundance of stroma. And indeed, Dr. Tang semi-quantified the immune and stromal infiltration, confirmed our classification. Indeed, she confirmed that the immune infiltration was way higher in the inflamed, and there was a progressive decrease. And then inflammatory stroma had the largest stromal deposition. Interesting, when we analyzed the immune composition of each class, we observed that the 3 non-inflamed, there are these 3, were enriched immunosuppressive elements. And in particular, the patistem-like was enriched in macrophages type 2, whereas Tregs were abundant in this class. We performed CD8 immunostaining, and we confirmed that there is high abundance of inflamed in the CD8 cells in these 2 classes. They are the inflamed, and the FOXP3, the Tregs, were more abundant here. Interesting, this was based on stroma and immune features, but still, the molecular features of the tumor seems to follow this classification. And we observed differential enrichment of a specific mutation. Again, I want to focus your attention on this class. That is the one that we call the patistem-like, this ICCA with stem cell features. And we observed enrichment in chromatin remodeler genes like ARID1A, BAP1. We also had IDH1 and 2 mutations that were enriched in here. And these have been described as the first gatekeepers of cholangiocarcinoma. We also had enrichment of AGFR2 fusion, but I have to say that when we looked at the histological characteristics of these tumors, we did not see any difference in terms of large or small bile ducts. When we looked at molecular pathways, interesting here, again, I want to focus your attention on this class. This was enriched in Notch, protein secretion, and I will show you here, there was significantly focal deletion. These are chromosomal deletions that are very small, that include very few genes, which are likely tumor suppressor. And indeed here we had the chromosome 14 was harboring just 1 gene. This is the sub1, which is a tumor suppressor that induces activation of YAP signaling, which has been associated with stem cell futures. Then, of course, we wanted to overlap this classification with others. Here we have at the top these 5 classes indicated by the different color boxes. And if you look again at the epithelium-like, when we overlapped with previous classes of cholangiocarcinoma with stem cell future, these are almost the same. But when we overlapped with HCC classes with stem cell future that are here at the bottom, look at the red color, which means presence and prediction for that class. There was no overlap whatsoever, suggesting that the ICCA with stem cell future have nothing to do in terms of molecular profile with the HCC with stem cell future. They're different entities. They have nothing to do, even if they seem to have some stem cell futures. Before going back, it seems that although, as I say, please do not take this as proof of origin, it just seems that an ICC with stem cell future may derive from a cholangiocyte precursor at different levels of maturation. And the same could happen with HCC with stem cell futures. They might derive from a hepatocyte precursor, but from the differentiation of an hepatocyte. But then what about mixed hepatocellular cholangiocarcinoma? Can we say anything in terms of molecular profile? Can we help in the classification of these tumors and the different subtypes? Do they represent a unique entity? Clearly, we know they do not. And unfortunately, in terms of mixed hepatocellular cholangiocarcinoma, we know way less than what we know for HCC and for cholangiocarcinoma. It's been extremely difficult to collect enough samples to conduct a study. We were extremely lucky here at Mount Sinai when we published this in 2017. And with the help of actually Dr. Tang, we were able to collect 18. And at the time, it was really an achievement to have enough of these mixed types to conduct enough material to perform chromosomal aberration, mutation profiling, and transcriptomic profiling. In this case, we had 18. We had 4 that were classical type. This is according to the WHO 2010 classification. We had also 14 with stem cell futures, including 6 that were cholangio. And we were really interested in understanding a little bit more if they are similar, what's the difference, what are the similarities and dissimilarities within this large subtype that I would say is kind of a spectrum, it's really different subtypes all in one. When we did transcriptomic profiling and we performed just an unsupervised analysis to identify which tumors look more alike, we observed that the cholangiocarcinoma, here in purple, seems to group all together in one different. You can see here the tree, this is all separate. On the other hand, we had, for the classical type, we were able to separate the HCC from the cholangiocarcinoma and perform the same analysis, and they seem to go together. On the other hand, what was a stem cell future seems to represent a different group, suggesting that they're not similar to each other. And in particular, we observed that the cholangiocarcinoma was very different from the others. We looked for hepatocytes markers, biliary markers. This is all gene expression. And here you can appreciate the immunohistochemistry. And the cholangiocarcinoma was the one that was very different from the others. They were clearly biliary-like expression in terms of RNA, but also they were clearly positive for CK7, CK19, EpCAM, and NCAM. But there was no expression of hepatocyte markers. On the other hand, if you move to the stem cell and the other classical type, they were kind of more biphenotypic, both at the transcription level and also at the, you can see here, there was a clear difference at the immunohistochemistry level. And in particular for the stem cell-like, although here the size of the cohort was very small, so I would like to advise some caution, we were able to identify 2 different groups according to the positivity of SALT4. And the SALT4 positive were biphenotypic in terms of gene expression, whereas though they were SALT4 negative, they were closer to an hepatocyte-like tumor. And finally, for the classical, again, we were able to separate and analyze separately. You have here the ICCA component and the HCC component. And these were very interested because at the gene expression level, they were biphenotypic. We will observe biliary markers and hepatocyte markers. But when we looked at the histological phenotype, if there was an ICCA, then they were only positive for CK7 and CK19, and the opposite for HCC. Although at the gene expression level, they expressed some biliary markers, when we looked at the histopathological profile, they were positive only for hepar and other markers of hepatocyte differentiation. We wanted to further investigate the similarity between these subtypes and classical HCC and cholangiocarcinoma. And we performed this large unsupervised clustering. You can see we have 327 tumors, some intrahepatic, some cholangiocarcinoma, and the 18 mixed. And once again, it was really interesting to see the cholangiocarcinoma. They went together with the cholangiocarcinoma. They had nothing to do with the HCC or with the other mixed tumors. Whereas when we look at the stem cell, hepatocellular cholangiocarcinoma, they were more similar to the HCC with stem cell features. Again, we only had 18 samples, therefore caution is advised, but actually, as I will show you later, another study confirmed most of our results. And the main message from our study is that definitely cholangiocarcinoma seems to be very different from the mixed tumors. You should not be considered mixed tumors, and actually seems to be more a biliary-like tumor. It was actually quite unique in terms of chromosomal stability, was quite stable in terms of chromosomal aberration, lower rate of mutations, and had an enrichment, TGF-beta signaling, and a specific immune response. Therefore, our main conclusion that cholangiocarcinoma is definitely a biliary-derived tumor. On the other hand, in terms of mixed tumors, there were tumors that were retained in both profiler hepatocyte and cholangiocytes, and in particular within the stem cell, the SALL4 positive were biphenotypic. On the other hand, the classical histological markers, again, they were quite distinct, but at gene expression level, they were biphenotypic, suggesting that indeed these mixed tumors, they may resemble more HCC or cholangiocarcinoma, although it's difficult to speculate about the cell of origin. Another study, this is actually the largest conducted thus far about mixed hepatocellular cholangiocarcinoma, confirmed most of our results, but in addition, added another level of complexity. In this case, these authors were able to collect 133, which is an impressive number of mixed tumors, and they had to recruit 8 different centers from China, Japan, and Singapore. Unfortunately, in this case, they didn't go by the WHO classification, but they used the Allen and Lisa classification. Therefore, the comparison is not straightforward. And you can see here, they included separate type, combined type, and mixed type. And we can debate if this was correct or not, but this is what they analyzed. And as you can see here, they have only six separate type, 56 combined type, but actually in some cases, they were able to perform microdissection and isolate the HCC component versus the cholangiocarcinoma component. And then they had 59 mixed type, which should refer to the stem cell features. Therefore, here you can see how they look like, and they perform a lot of next generation sequencing analysis. You can see here they perform whole exome sequencing to identify mutation, whole genome sequencing, RNA-seq, and so on. And what they observed is that indeed mixed tumors are kind of like their own entity. In particular, in terms of mutations, they observed significant differences compared with HCC and cholangiocarcinoma. For example, TP53 was way more mutated in mixed hepatocellular cholangiocarcinoma tumors, something we had already observed in our study, but with 18, it was difficult to make a strong conclusion. They also observed the mutations that are typical of HCC, like TERT and CTNB1, were way lower in mixed hepatocellular cholangiocarcinoma. And also KRAS, which usually observed in 25% of intrahepatic cholangiocarcinoma, was absent in mixed hepatocellular cholangiocarcinoma, suggesting again that the mutation profile seems to be kind of not intermediate, but no one or quite the other. And actually, they also identify specific drivers for mixed tumors, and here I've listed a few, which seems to suggest that, again, mixed tumors should be considered separate, a different entity. What about transcriptomic profiling? Are the mixed tumors closer to HCC, to cholangio, and can we say something about their cell of origin? More importantly, similar to what we did before, they merged their mixed tumors with HCC that are here indicated in orange, and with cholangiocarcinoma that are here indicated in green. And when they performed an unsupervised clustering, they identified 4 different groups. And interesting, as you can see here, the differentiation was different from one group to the other. In particular, they found that this group called P1 had a biliary differentiation. And indeed, if you look at the color, here we had mostly intrahepatic cholangiocarcinoma with some mixed tumors. The P2 was poorly differentiated, and here they had mostly HCC with stem cell features and actually mixed tumors. And then moving on to the P3 and the P4, these are mostly, as you can see, they are all HCC with actually more than hepatic differentiation. But what about the mixed tumors? Are they all the same, equally distributed in P1 or P2, or there is a difference? And interestingly, they observed that they combined, where they actually were able to separate the 2 different entities, those were closer to cholangiocarcinoma. They all grouped together here. It's clearly by these color-coded boxes. On the other hand, something that we had already observed in our study, those with mixed with stem cell features, those were closer to HCC with stem cell feature, the classical HCC with CK19 positive. The last thing of their study, which I find is like one of the most perhaps debatable and more interesting, is that also by using the mutation, the chromosomal aberration, they try to assess which of those tumors derive from the same cell or a different one, if they had a monoclonal or a polyclonal origin. And unfortunately, most of this was done in the separate cases, but interesting, they observed that in most of cases, except these two, where you can see that these circles do not overlap, in the majority of cases, actually, those tumors derive from the same cell, because they share the large amount of mutations and chromosomal aberrations. Therefore, going back to the highly debatable figure, we have classical ICCA derived from cholangiocytes, classical HCC, perhaps deriving from an hepatocyte, HCC with stem cell future, very different and very far from the ICCA with stem cell future, and the cholangiola, which should be aligned more with a biliary-like tumor. So what about the mix? Of course, they are in the middle. We can now speculate to which cell is actually giving rise to these tumors, but I think that there are multiple possibilities that hopefully will be clarified in the next studies. And I think that all of these will actually arise. But what is important is that most of these mixed tumors seem to derive from one cell. Therefore, to conclude, molecular profiling of liver tumors over the years has provided novel insight into the molecular pathogenesis and hopefully has helped in understanding the different subtypes and the complexity of this spectrum. Of course, we have also used this data to speculate about potential cell of origin, which unfortunately are just a speculation. We cannot, without functional studies, confirm our speculations. In our case, in our group, we have focused on generating a novel classification of intrahepatic cholangiocarcinoma. And to my surprise, I have to admit, we have identified these largest non-inflamed classes. There is an ICCA with stem cell futures, and again, I refer to the molecular profile. They have a molecular gene expression profile of stem cell futures, although these are pure intrahepatic cholangiocarcinoma. And we're currently further exploring the implication of this. Subtypes of HCC and ICCA with stem cell futures have been identified and confirmed in multiple studies. And they're very different from each other. They have different origin potentially in the same molecular profile. We have distinct subtypes of mixed tumors. Sorry, here is mixed tumors could be identified for mixed with both mono and multiclonal origins. Cholangiocarcinoma, at least at the molecular profile, I would be interested in hearing your opinion, is definitely a molecularly differentiated entity of cholangiocarcinoma. And my hope for the futures is that single cell sequencing studies will be performed in large cohorts of HCC, cholangio, and mixed tumors. And this should be conducted to try to gather a deeper understanding of which is the definite cell of origin in liver cancer, assuming that there is one. In reality, we expect more than one in a complex scenario. Finally, I would like to thank all my colleagues, Dr. Swan Tang. Without her, we couldn't be able to conduct any study here in Mount Sinai. I have to say she's a pillar of all our studies and current, previous, and future publication. And thank you all for your attention. I'm happy to take any question.
Thank you very much. Yeah, I'm back. I lost some connection so I had to restart my computer. It took some time. My excuses for that. Yeah, it's very, very intriguing, of course. But yeah, sorry, maybe I missed it. But is there a marker for your non-inflamed stem cell intrahepatic cholangiocarcinoma? Is that the same with the SAL4+, or am I really mixing it all up? Because it's quite...
We haven't checked the SAL4. We definitely use the different molecular profile in terms of transcription. Transcription level, we see a lot of transcription factors. We haven't really moved on to validating if we can identify a specific marker, but that is the next step. As I said, I was really skeptical in the beginning of this ICCA with stem cell futures. I saw it myself in our data set where we know the origin, that we know that they are pure cholangiocarcinoma, because with the other studies, it was perhaps there is a misdiagnosis, there are mixed tumors, there are not really cholangiocarcinoma. But we're really interested in understanding what it means, if it really can reflect a different cell of origin, although again, it's highly debatable how we can prove that. But we're actually looking for markers. Yes, we're actually looking for biomarkers for each one of these classes, since we actually have associated each of these classes with specific therapies. And we have validated this in murine models. I didn't have time to show all the data, but yeah, we're interested in looking for markers. They could help identifying the class without need to access the RNA and perform profiling, which, as we know, it's very difficult, especially with advanced cases.
Yeah, well, in the clinics, it's of course not so easy to go immediately for. Okay, so Are there still other questions from the floor? Otherwise, maybe we should proceed. Or should we now first go for your explanation of the journal, Giuliano, or do we do that on the end? What do you think? Maybe we should do a small break just for people to be able to go to the bathroom or so.
Okay, sure. Sure, we should 5-minute break.
Yeah, 5-minute break, and then you explain the
And
journal.
then I will give a short, you know, I was asked to present
Yeah, but
few
it's
items
important.
of the journal, and yeah, okay. Let's make
Yeah,
first
so
of all,
let's meet again.
thank you for the nice talk, Daniela. I would ask a lot of questions, but you know we don't have time. Thanks a lot.
You can email me. Happy to
Yeah.